So good afternoon, everybody. And for any of you who are on this call that don't know me, I am Susan Carpenter and I'm an assistant professor in MCD biology. And I'm an immunologist, and my area of research is our innate or our first responses of the immune system. And so a disclaimer is that I am not a researcher who studies adaptive immunity and memory. However, I am really delighted to have the opportunity to talk today with and have my co-presenter be Professor Rebecca DuBois, who's Associate Professor in Biomolecular Engineering. And Rebecca's area of research is in she studies many viruses, including astroviruses, RSV, influenza, and she studies them from the angle of their molecular mechanisms, understand their structures to be able to better develop vaccines. And so while she does not also work on SARS-CoV-2, she has a lot more knowledge when it comes to vaccine development. So between the two of us, we're hoping to use our knowledge today to perhaps tell you some things maybe that you're not aware of, or and give you our perspectives from our areas of expertise. Okay, so let's get started. So our overview of what we're going to go through today is that I am going to give you an immunology 101. So hopefully a revision for everybody on how our immune systems function, in particular how they're functioning as it pertains to SARS-CoV-2 infection and also in vaccine, how they respond in terms of vaccines. Then Rebecca's going to take over and she is going to talk to you about the vaccines that are currently available, give you some insights into how they're working. And then we are going to come back and circle back to what vaccination is going to mean and how we see ourselves returning somewhat to normal life in the near future, and we're going to answer your questions. And throughout today's talk, when I'm talking, Rebecca's going to monitor the chat, and then I'll monitor the chat for when Rebecca's speaking. So feel free, if you have any additional questions as we're going along, you can put them in the chat and we can answer them as we're going along. And I thank everyone who filled in the survey before the presentation, and we kind of tried to gear our presentation around answering some of your questions that you have. So hopefully we'll answer your questions as we're going along, but also at the end we'll save time so that we can address any additional questions that we might not cover. Okay, so Immunology 101. As a reminder to everyone, when we're talking about our immune system, as I said, it can be divided into two halves. You have innate immunity and adaptive immunity. And so your innate immune system is that first line of defense. It's the one that gets activated within minutes to hours when it encounters a microbe. And this is the area of research that my lab particularly focuses on. And so when you activate your innate immune system, so here you have a pathogen that's entering and going to be activating cells of the innate immune system that will trigger these pattern recognition receptors to initiate these signaling events that are going to turn on inflammation. And so we know that inflammation in a transient sense is critical to keep us protected against microbial infections. So we're going to turn on these cytokines and interferons that are really going to shape the downstream activation of your adaptive immune system. So the adaptive immune system is that later response, highly specific, and this is what gives you memory. So activation of your T and B cells is what is going to help overall clear the pathogen and provide you with the memory. And so this is the part of the immune system that we're really targeting when we're targeting with vaccination. And what I'm going to walk you through in terms of the immune response, I want to acknowledge first Dr. Akiko Iwasaki, who is an amazing immunologist and virologist. She works specifically on SARS-CoV-2, and she developed this really cool tutorial in collaboration with BioRender, walking people through the immune responses. And so I'm borrowing her slides. And so if anyone's interested, I highly recommend checking out Dr. Iwasaki's work. And also she's very active on Twitter and is just a great rational voice out there for all things that are happening in relation to SARS-CoV-2. Okay, so we are here today to talk about SARS-CoV-2, which is the virus that is the causative agent of COVID-19. And so here we're looking at SARS-CoV-2 as it's entering cells. This is a EM stain. And then in terms of the cartoon image of what we're talking about, here is your SARS-CoV-2 virus, which is a single-stranded RNA virus. And it is studded on the outside, with the protein they call the spike protein, which is going to be mostly what we're talking about today. Because the spike protein is critical in that this is the protein that allows the virus to gain entry into cells. And it does this through binding through the ACE2 receptor on the host cells. And this, of course, is the target for vaccine development. And so once the virus gains entry through the ACE2 receptor, what happens is you start triggering off that innate immune response. So here we have the virus getting into those cells and triggering the production of type 1 interferons. And type 1 interferons are the critical proteins to try and protect you against viral infection. So they get their name interferon from interfering with viral replication. And so it does this in a number of different ways. One, the interferons can signal to neighboring cells to warn them of the fact that there is a virus in the body and put up barriers to try and protect themselves. Interferons can also trigger many other proteins and in fact signal for the cells to die, so that you can eliminate the microbe by this mechanism. And third, what interferons can do is they can help recruit additional white blood cells that are going to stimulate the immune system to provide that long-lasting memory. So this is where your interferons are going to play that big role in activating the adaptive immune response. And here is what we're going to cover, a few of the steps that are critical to understanding both the virus and how we vaccinate. So viruses are going to infect and replicate, and the SARS-CoV-2 is infecting your epithelial cells. And then these cells can then activate your innate immune cells, in particular dendritic cells. Dendritic cells represent, they're known as professional antigen presenting cells, because it's their job to spot the infection at the site where it's infecting and then become activated and move to the nearest lymph node to alert and find the adaptive immune response cells in order to kick in that long-lasting response. So your dendritic cells are going to move to the lymph nodes where they're going to encounter and find and present the antigen to T cells, which are going to become activated and help activate your B cells. And what their job is to then move back to the site of infection and basically function to produce, your B cells are going to produce your antibodies, and your T cells are going to help to kill those virally infected cells. And so this is just kind of giving you a more insight into those responses. So here you have your antigen presenting cells, your APCs, your dendritic cells. They are really critical in activating the T cell response. And what we're showing you here are your CD8 positive T cells, also known as your cytotoxic T cells. These play a huge role in viral infection because they can then go to the virally infected cell, recognize the antigen being presented, and kill these cells through apoptosis. Your antigen presenting cells are also going to activate the class of T cells known as CD4 T cells that play big roles as helpers in all of these responses. And we're going to help to turn on and activate your B cells. And one of the major roles of your B cells are, these are the ones that are going to pump out the immunoglobulin. They're going to pump out antibodies that are going to do multiple things. But one of the major role of these antibodies are as neutralizers. You need to produce these neutralizing antibodies to bind to the virus and stop it from entering cells. And so we know that in about 80% of cases for SARS-CoV-2 or COVID-19 is that people do really well and they're asymptomatic or they clear the infection. However, we see in a number of cases that there can be severe complications, and this is due to an imbalanced immune response. And so one of the things that we see in severe COVID cases is this idea of activation of the cytokine storm. And what's happening here is that viruses that are now making their way through the respiratory tract and infecting lower levels of the lung, here we're going to activate additional immune cells like macrophages, and they are going to kick off a continued induction of inflammation. And so you're going to have tons of these cytokines that are going to be produced. And when this is out of control and is not being stopped in the transient manner that we just described, what happens is it continues to attract more and more immune cells. And so what this does is it leads to weakened blood vessels that are going to allow more fluid to accumulate within the lung. We're going to see damage from deposits of fibrin. And together, what this results in is that this can result in respiratory failure. And so sometimes I get asked the question that if there's so many asymptomatic cases, is it better to have natural infection versus vaccination? And so I would absolutely say that no, we do not want natural infection over vaccination for a number of reasons. So when we develop vaccines to protect us against these infections, they're developed in such a way that you're trying to optimize the immune response. So you're trying to produce a much more robust as well as a much more long-lasting immunity that we see over natural infection. And the only way to achieve herd immunity in a safe way is to vaccinate. And as we know, we are going to be required to get between 70 to 85% of people vaccinated in order to hit herd immunity, which we will touch upon a little bit later in today's lecture. And the vaccine does a number of things. Not only is it going to protect you against the particular virus, but it's also going to protect those around you. So if you get a natural infection, you're infectious, you're contagious, and you can impact those around you. And so while you might be asymptomatic, you know, the other person or people in your house or wherever you encounter, they might not do so well. And so vaccines are not just for you, but they're for protecting those around you. Also, what we see with SARS-CoV-2 is that, like all viruses, they are clever in many ways, and they have many ways of evading the immune response. And so they can do things like attempt to subvert that type 1 interferon response. Many of the NSP proteins within the virus are designed specifically to target those innate immune signaling pathways to mess with the type 1 interferon response. And so we see that a delay in that response can have serious consequences to the host and lead to some of those severe cases where we see then later out of control inflammation. And we know that the risk of complications from the virus are much, much higher than what we're seeing any safety concerns surrounding the vaccine. So the risk of getting the virus and you don't know how you're going to respond, this is much, much greater than taking the vaccine and protecting yourself.
Susan, I'll just jump in real quick. Marm Kilpatrick made a comment that, you mentioned that approximately 80% of cases COVID-19 is not severe. And he clarified too that the fraction of infections that are asymptomatic is approximately 20%.
Great, thanks for that, Marm. Okay, and so here really what I'm just highlighting for you are the principles of immunity. And these principles of immunity are both the same for exposure to a virus as they are to obtaining a vaccine. And so initially, this initial exposure or vaccination is going to initiate this primary immune response. It's where you kick off your innate immune system that kicks in within minutes and hours, that's sustained for days to activate that adaptive immune system. And what then we see is that if you have a secondary exposure or you meet the same virus again, or in cases where you're getting a booster shot of a vaccine, you get a much faster, much more robust, much more specific response the second time round. And so this is really the important features of how your immune system functions. And so just as a reminder, if you're getting the vaccine, the same principles apply as what I just walked through in previous slides. So if you're getting a vaccine, what you're doing is you're trying to activate those antigen presenting cells, your dendritic cells, which are going to take that antigen, which, as Rebecca is going to walk you through, is the spike protein. It's going to move to the nearest lymph node and initiate activation of those T and B cell responses to essentially provide you then with that long-term memory, so that the next time you might encounter, in this case, you'll be vaccinated if you encounter the actual virus, that you were going to be protected. And I'm going to hand it over to Rebecca, who's going to walk you through now what's happening with our vaccines that are available.
Great, thank you, Susan. Okay, so now I'm going to talk about the design and development of COVID-19 vaccines, and I will just start by highlighting that when this emerged, we had no previous established or licensed platform to make a coronavirus vaccine. So what has happened over the past year is that there has been manufacturing and testing of multiple different technologies or platforms in parallel. And every single one of these has required extensive safety testing in preclinical and clinical trials. I'll talk about those. And safety is utmost importance because vaccines are given to healthy individuals. And ideally, we would develop a vaccine that elicits better immunity than natural infection induces. We know that immunity to other coronaviruses that cause the common cold, and even immunity to this original SARS, SARS-1, that immunity wanes over years. So we would ideally like a vaccine that elicits long-term immunity. Next slide.
All right.
So these were the main sort of technologies that were initiated at the onset of the pandemic. And on the left are 4 that are existing licensed technologies where we have existing licensed vaccines made with those platforms. Specifically, the more traditional ones, the inactivated virus or a live attenuated, as well as recombinant protein. And then the new vaccine platforms include mRNA that we've heard a lot about, as well as fairly new would be the viral vectored and DNA. And so I won't talk much, or I won't talk at all about DNA vaccines because those really haven't emerged as sort of a, near-term solution, as well as live attenuated, which is always challenged with the fact or the balance of finding immunity but no symptoms. So I'll be focused on the other 4 technologies. So one thing you might not know about coronavirus infection is how the spike protein works to induce infection. And so if you want to play this, so this is a really fantastic video here on YouTube. So we have the spike glycoprotein, and you've heard a lot about that. And what I want to emphasize is that it's in a distinct conformation on the surface of the virus that allows it to bind to the ACE2 receptor. And this triggers a dramatic conformational change in that spike protein. And it's thought that multiple spike proteins then work together. I'm not sure why the video stopped.
Oh, I still see it working.
Okay.
Sorry.
It's not working right, but those proteins work together to fuse the virus and host cell membranes. And what you get after that fusion event is what's called the post-fusion conformation of the spike protein. And so what we want is an antibody response to the pre-fusion conformation of that protein. But as you can imagine, it is like a set mousetrap. It is in this metastable sort of relatively high energy state, and that can complicate vaccine development. And I'll talk about that in the next slide here. So this was published in November, but it was a concern that a lot of scientists that know about these membrane fusion proteins and their sort of metastable state we're concerned about. And this is sort of what you might get if you do make a traditional inactivated virus vaccine. And so the process of the chemical process of inactivation can trigger the spike proteins to undergo that conformational change. And so this publication shows evidence, cryo-EM evidence. And so you can see that a typical inactivated virus might have some copies of the pre-fusion form, which is what we want the immunity to, but a lot of the spikes have been converted to the post-fusion form, which we don't want immunity to. So this is a challenge in making vaccines against the spike, is we want to have immunity to that pre-fusion form, but it's metastable and kind of high energy. But some protein engineering has helped to overcome this issue. And this is a publication from 2017 where a way to basically lock the protein in the pre-fusion form was discovered. And that's by introducing some prolines. And if you remember your amino acids, prolines have this really rigid structure that minimize flexibility. And they were introduced at this key point where these helices are the ones that spring out to trigger membrane fusion. And they show by locking this little spring mechanism, locking it down, you lock the protein in the pre-fusion conformation. And so this strategy, this 2 proline mutation, is actually in most of our vaccine candidates right now, in our vaccines, including the mRNA vaccines. You'll see 2 consecutive proline substitutions. And also this was used to solve the first structure of the coronavirus spike. And so we'll go through the different vaccines that have been developed and how they work. First, we'll talk about the mRNA vaccines, which includes the Moderna and Pfizer vaccines. So on the left is just your typical SARS coronavirus 2 virion. The virus has many genes in it, including one that encodes the spike. And so an mRNA vaccine is very simple. It is just the spike encoded on an mRNA molecule. And this mRNA is encapsulated in a lipid nanoparticle. And we'll talk a little bit more about that in a second. So when you receive an mRNA vaccine, these nanoparticles are able to fuse with our own cells in our body. And that allows uptake of the mRNA inside our cells. And this mRNA gets translated by our ribosomes into the spike protein. This protein gets displayed, secreted and displayed on the surface of that cell. And also this protein gets chewed up into fragments, and those fragments get displayed. And that's important for activation of different immune cells. And I'll talk about that in a second. So if you look at the mRNA vaccine ingredients, while it looks like a long list, it's really quite simple. It's your mRNA that encodes the spike protein, the lipids that encapsulate the mRNA, as well as some salts and sugars and buffers. And so there are slight differences in the lipids that are used to encapsulate the mRNA, but really the mRNA is very similar and encodes the full-length spike glycoprotein with those 2 proline mutations. We know there are some differences in sort of the amount per dose and sort of the needs for freezing. We've heard a lot about how the Pfizer vaccine needs ultra freezers, whereas the Moderna doesn't. And that's probably something to do with the formulation of the mRNA loaded lipid nanoparticles that is related to their vaccine stability as well as efficacy. And most people in this audience, in the RNA world especially, know how fragile RNA is, and so that is likely the main reason why these need to be frozen and kept frozen just to maintain the stability of that mRNA. The next vaccine I'll talk about are the adenovirus vectored vaccines. So adenoviruses are very common viruses that often cause sort of common colds and mild illness in people. And so an adenovirus vectored vaccine is using adenovirus to carry the spike coronavirus gene encoded as DNA inside this non-replicating adenovirus. So this is essentially a little package that carries the spike gene. And so this virus can infect a cell, meaning it can enter and release that DNA into the cell. However, the virus itself, the adenovirus, cannot replicate. It is replication deficient. So that DNA is introduced into our nucleus, and that DNA gets translated into mRNA. And at that point, it's very similar to the mRNA vaccines. The mRNA gets translated to spike protein, which gets displayed on the surface of the cell. And finally, I'll talk about the recombinant spike protein vaccines, such as that made by Novavax, which is in phase 3 clinical trials right now. And so this is a fairly well-established vaccine technology where purified recombinant SARS coronavirus spike protein is produced in the lab and purified. And it's purified on little lipid nanoparticles, sort of shown here with multiple spikes on them, but this is just the purified antigen protein. And because this is different from the other vaccines, it requires an adjuvant shown by these little gray circles here. And the adjuvant just sort of helps to stimulate our immune response to the spike protein in the vaccine. And so just to remind you, going back to what Susan was talking about, all of these vaccines are intended to elicit antibodies. And the way that happens is that antigen presenting cells are going to uptake and display the spike protein that is produced or introduced during vaccination. And this will help helper T cells, which will help to activate our B cells. Those B cells, the ones that make antibodies that recognize the spike protein, will be activated to produce antibodies. And those antibodies will exist in our bloodstream and be ready to stick to the coronavirus and prevent it from causing an infection. And so these same antigen presenting cells will also activate killer T cells or cytotoxic T cells. And these cells will be able to recognize any infected human cell that's displaying pieces of the coronavirus spike protein. So having these cytotoxic or killer T cells present are another line of defense if we happen to get one coronavirus that goes past our antibody response. Hopefully the killer T cells will be there as a backup and be like, okay, I will get rid of this infected cell. Okay, so now I'd like to talk about the vaccine clinical trials, and this is sort of a timeline. And the main thing I want to emphasize is that a lot of concern about the timeline of vaccine development. I want to alleviate those concerns and assure you that full clinical trials were performed, and I'll explain that in more detail. But immediately in January, the design of vaccines was started as soon as we had the sequence of the SARS coronavirus 2 genome. And phase 1 clinical trials were started in April. These are very small. They're intended to test safety and perhaps different doses or slightly different antigens. And phase 2 is a little bit larger to evaluate immune responses. And phase 3 is where you evaluate efficacy. And so these were not fast-tracked. Phase 3 was a completely normal phase 3. What was fast-tracked was the production of the vaccines. Normally you wouldn't start production until you know that the vaccine works and will be approved, but production was done at risk, and this was the fast track of our vaccine development, producing vaccines that may not make it all the way through to FDA approval. So I will just show one data slide. This is from phase 2 clinical trials of an mRNA vaccine. What you can see is the amount of neutralizing antibodies. And so this is drawing someone's blood and asking, do they have antibodies that block virus infection in the lab? And so of course, never infected people don't have neutralizing antibodies. This shows natural infection from COVID-19, and you could see a real variety of different levels of responses to natural infection. And then what you'll see in the next 2 panels is response to by vaccinated individuals, either they're a little bit younger or 65 and older. And so on day 1, there was no neutralizing antibodies, but 21 days after the first dose, you can see that there was a neutralizing antibody response, but a second dose really boosts the levels of neutralizing antibodies in individuals. And you can see it's really very, very similar in terms of the antibody responses, even in over 65 individuals. And what I want to emphasize also, again, is that you can see that this vaccination response really exceeds that from natural infection. And so a lot of people ask, why are we giving booster shots? I think that data shows that, but also just sort of what you would see here is your antibody concentrations of different kinds of antibodies, as well as the affinity, how tightly they stick to the pathogen, the virus of interest. And so after your first dose, you will get your sort of primary IgM response, and then your second dose will stimulate higher levels of IgG response. And those are your more specific antibodies. These are higher affinity antibodies. You get higher concentrations and ultimately longer lasting antibody levels after a second dose. So here's an overview of the 2 mRNA vaccine clinical trials. I want to emphasize they were blinded, meaning neither the individuals that were in the clinical trial nor the doctors knew whether they were getting placebo or the vaccine. And so they both had thousands of volunteers split in half. And after the vaccination, we had to wait. We had to wait and observe and see what happens in terms of natural infection in these individuals. And so you can see they sort of waited for a certain number of cases to occur in their whole group of vaccine volunteers. And then that was unblinded. And they said how many people got the placebo versus how many got, oh, I'm sorry, this is a typo. This is how many got the vaccine. And same here, the placebo versus the vaccine. And what you can see here and a little bit more clearly in this slide is that this is the incidence of COVID-19 infection in vaccinated individuals versus placebo. And you just see increased numbers of infection in placebo. So the vaccine really clearly shows protection and efficacy. I'll note also in the Pfizer one, you can see the little purple arrows are patients that have severe COVID. And so the vaccines really clearly show protection against severe COVID too. And finally, I'll just note that there are many excellent female scientists that have really played major roles in development of the mRNA and adenovirus vaccines, and wanted to give a shout out to them.
Rebecca, just a couple of things maybe before I take over. One clarification point, I think we had an error in one of our slides that the Pfizer vaccine is the one that's stored at -70, -80, and Moderna is -20. I think we had it the other way around.
Oh, I'm sorry about
that. I see some questions about the adenovirus and the types of serotypes. I don't know, do you have any information on that or anything you want to add?
Yeah, every company has really chosen a different adenovirus because these are such common viruses. People, scientists have been on the hunt for these rare adenoviruses where most of the population does not have any pre-existing immunity to them, which would make these viruses amenable to an adenovirus vectored vaccine. So you don't want to have pre-existing immunity to them, and many of them have chosen different adenovirus strains as their sort of vector of choice.
And I'm just trying, there's a lot of questions, so I'm just trying to maybe compile a few together. We're asked, what's in the placebo? Is it delivery vehicle or just buffer?
That's a great question. I believe it's just buffer.
And then just to clarify, the Johnson & Johnson is also a DNA adenovirus based vaccine.
Yes.
And do the clinical trial data report on presentation of symptoms or DNA evidence of infection?
The clinical trials are for COVID-19 symptoms, no matter how severe. So I would say that, you know, any kind of increase, this is seen as sort of a failed protection of the vaccine, but many of these cases are known to be mild cases. You know, the rare ones that you do see in vaccinated individuals are known to be mild. So it's not really fully representative of how protective the vaccine is, but what was the primary endpoint was protection from COVID-19 disease.
Okay. And Marm to the rescue again. Marm says that the placebo is just saline. So thanks, Marm. And this is why also have experts who know more about these things on the call to help us get the right info out there.
Right.
Okay. Okay, so we're going to spend just, I'm going to come back in for a couple of minutes to talk a little bit about efficacy and side effects. And really what we're seeing in terms of efficacy varies from vaccine to vaccine. So the Pfizer and Moderna are very similar, where we see this efficacy of about 95%. Johnson & Johnson's is at about 66%. AstraZeneca, for a number of reasons that we don't have time to spend talking too much about today, but they had a miscalculation of some of the ways in which they administer the vaccine. And so they had this efficacy range of between 60 and 90%. Novavax is around 89%, and Russia's Sputnik is at about 92%. But I think the real take-home message here is that, importantly, most of these vaccines, what they're doing is really preventing severe disease. And this is going to play a big role in terms of allowing our hospitals to stay effective and not overwhelm the system. And I just want to point out quickly that the Johnson & Johnson vaccine is a single-dose vaccine. And so they are currently trialing giving a booster dose in the hopes that that would get them up to the range of the 95s that we're seeing with the vaccines like Moderna and Pfizer that are 2 shots. Because as Rebecca says, when you give a second dose, you get an even stronger and more specific immune response second time round. So that's underway right now. Next slide. And so we've gotten some questions on how exactly is efficacy calculated? And so while this is very wordy, really what it's getting at is the equation that we see in the middle. So vaccine efficacy is measured by just calculating the risk of disease among vaccinated versus unvaccinated individuals and determine the percentage reduction in the risk for the disease among those that were vaccinated, comparing them to the unvaccinated. So the greater the percentage of reduction of illness in the vaccinated group suggests better efficacy. So simply the risk ratio, if you click next, it's a very simple equation. And what we've seen from the clinical trials for Pfizer is that they recruited about 43,000 individuals. They waited for about 170 to become ill. And then they see that out of the 170, 162 had received the placebo shot, 8 had received the real vaccine. And it's a very simple equation and that's what gets the 95%. If nobody fell ill that were in the vaccine group, that would equate to 100% efficacy for a vaccine. And I just think it's worth pointing out the difference between efficacy versus effectiveness. Efficacy is really the term that's used in a clinical trial setting. And remember that a clinical trial setting is that perfect setting, right? They're selecting individuals who don't have any comorbidities or within a particular age range. And so the efficacy from a clinical trial might end up being different to the effectiveness that we see out in the real world, because that effectiveness, we're going to use the vaccine on individuals that would not enter into a clinical trial. So it's worth noting that that's where those differences can come out in the real world. Next slide. And we want to just spend a brief moment touching upon the variants that we're seeing, because this does play a big role in how our immune response deals with the virus and also how effective our vaccines are going to be. So we are seeing in particular these 3 main variants that have emerged in recent months. So the B1.1.7 is the UK strain, 1.351 is the South African strain, and P.1 is the strain that's been seen out of Brazil. And what this complicated slide is showing you really is that there is a number of different mutations occurring. And obviously the ones that concern us most are those that are within the spike protein. So if you go to the next slide, it kind of just breaks down what we're seeing more specifically between the 3 variants. And so you can see there's, over 20 mutations around in each of these different strains. Many of them, about half of them within the spike, and the rest are throughout the different parts of the virus. And this is emerging. The reason why we called this seminar What We Know So Far is that we could probably come back in a few months to give you an updated version of this, because this is really a moving target. And so there's not a lot of data out yet on how these variants are impacting us. But what we're seeing so far is concerning in that transmissibility, particularly for the UK version, is over 50% increased. And so that's why this is becoming the dominant strain in many countries. And I think it's estimated that it's going to be the dominant strain throughout the US by March. And we don't know yet with the other 2 variants, there's not enough data out there. Also, in terms of the lethality, again, there's still not enough data. What we're seeing is that some of the later vaccines that are going through trials have included some data on some of these variants, but it's worth noting that the Pfizer and Moderna trials were done before these variants came out, and so that could impact. how effective they are. So it's still an ongoing area of research as to the impact that these strains are going to have on the effectiveness of in terms of the vaccines, because remember, the vaccines are developed around the spike protein, and we're pretty confident that you're still going to have quite a bit of protection, because remember, spike protein is a big protein that we're making many antibodies to, and so we're hoping that many of them will still be effective. And so there is some data on this specifically from the Johnson & Johnson trial. And so here's just some breakdown that we see. The Johnson & Johnson vaccine is 72% protective in the US, 66% in South Africa, and South America, and 57% in South Africa. And in South Africa, that the major variant that was present is that current variant that we're seeing. And so we think that's what's bringing down the protection level, but we are still seeing protection. And remember, this is only a one-shot vaccine, and we hope that that will be better if it's used in a two-shot. So while there is some concern, we still believe that our current vaccines will give significant levels of protection. Next slide. Okay, so in terms of side effects, many people have been vaccinated already. So even within the US, 48 million people have received one dose. That's over 10% of the population, and about just over exactly 3.4% have received 2 doses. So that's a lot of people that have been vaccinated. And so really what we're seeing in terms of side effects are some in terms of immune response. The most concerning are the allergic responses. So there have been some anaphylactic-like responses, and it's about between 2 to 6 cases per million. And the good news is that all individuals were able to be treated using an EpiPen, and there's a lot of research trying to understand why exactly it is that we see some of these allergic responses. Some were in individuals who had previous allergies to other types of vaccines, but some weren't. And so there's investigations into, is it the lipids, is it the PEG that's used in the vaccines that could be impacting these immune responses. But again, these are very small numbers, so it's a very, very rare occurrence. And that's why at all vaccination sites, they have EpiPens, and these allergic responses happen within minutes. And so they're very quick to be spotted. There's been some data on Bell's palsy, but that data suggests actually that it's no more common than is seen in general in the population. But there's, with vaccines, particularly the second shot, you can see more fever and more of those impacts that you're feeling quite. You might have a day or two of feeling like you have flu-like symptoms, but I would just emphasize that that's your immune response, hopefully doing its thing and keeping you protected. Okay, and we have some data that really is suggesting that vaccination is working. And so this is data from Israel. And my go-to source is Dr. Aaron Siegel, who's at the Weizmann Institute. He posts this info on Twitter. So disclaimer, it's not peer-reviewed, but this is the current data that's coming out of Israel, where they have done a really high-level number of vaccinations. So they have over 80% of over 60s have now been vaccinated. And so this data is as of this week, where they're seeing that 3 weeks after starting the second dose administration, what's really nice is that they can see in that population, the over 60 population, you're seeing this downward trend in critically ill. And so they're seeing 56% fewer cases, 42% fewer hospitalizations, 34% fewer critically ill, and 35% fewer deaths. So this is giving us hope that this vaccination is working. And that is just that's the graph for the hospitalization. So you can see in the plus 60s after you're seeing, like just in the last couple of weeks, that significant drop in the number of hospitalizations. So this is really promising data. And we have some suggestions from data that's coming out of the AstraZeneca trials, is that suggesting that there is a reduction in transmission, which is also obviously going to be very important to try and help stop this the reservoir of the virus. And so I think this data is also really encouraging, as well as the fact that their first single dose of the AstraZeneca vaccine looks like the same level of efficacy that we're seeing out of the Johnson & Johnson. So even after that first shot, we're seeing quite a bit of protection, and that second shot is going to help with the booster. And so far, there's promising data in relation to the Moderna vaccine that's been done on primates that are showing that vaccinated monkeys really help clear the virus if they encounter it. And so I think, again, all of this data is suggesting that, as we would hope, vaccines are doing their job and will help curb the viral infections. Next slide.
A quick point of question: how do we know how transmission was measured in the AstraZeneca data?
I can't tell you off the top of my head, but yes, we can put the link to that paper, what exactly they're measuring for transmission. It's a good point. Okay, and just again, myself and Rebecca are here to promote vaccines, and we cannot wait to get vaccinated. And I think this slide just really, again, reminds us all that vaccine programs really save lives. So I know this is a super busy slide, but what I want you just to focus in on is the fact that those big blue circles are the ones that are highlighting the active disease. And if Rebecca can just highlight measles there, you can see that where the orange dot is when the vaccination program started for measles, and you can see what an impact it had on basically really massively reducing measles. But what we see is that if you become complacent about this and there's a drop in vaccine uptake rate, what happens is you see the reemergence of these viruses. And so you know, there has been an uptick in measles because of the fact that measles is super highly contagious. You need at least over 85%, even more, 90% of individuals to be vaccinated to keep the level of protection. And so when it falls below that, we start seeing this reemergence, and as well as cases. And we see, for example, you know, one of the biggest achievements of the vaccine programs is the eradication of smallpox. So in the 60s, there was a very active program specifically for targeting and vaccinating against smallpox. And we see that by the 80s, this was fully eradicated. So again, highlighting how important vaccines are to protect against various diseases. Okay, so how do we get back to a new level of normal, right? And there's multiple things, but really vaccination is going to be our major way in order to get back to life as we know it, as we knew it more than over a year ago. And so, but right now we know that there's a shortage of vaccines and we need the entire world to get vaccinated. And so there is still a major need for, until we can hit that herd immunity and limit the reservoir of virus, we are going to have to continue our social distancing, as well as continuing masking up and to try and protect ourselves while this vaccine program is being rolled out. And here at UCSC, I just want to point out some of the things that we are trying to actively do to get ourselves back into in-person work in general. And, you know, big shout out to the molecular diagnostics lab that have done an amazing job at getting us to be able to have testing continuously with a really, really quick turnaround. And Rebecca's lab has done great work in serological testing. They developed a test that you can determine if you have antibodies to COVID in like 15 minutes, right, from tiny drops of blood. And this I think is, you know, a crucial cog in our wheel of protection, because you know, there's thoughts now about if we had much better in place serological testing and you knew that you had some immunity, perhaps you only need one dose of a vaccine to be protected. And so this could be a way of helping speed up getting us to herd immunity, is by having good continuous serological testing. Also, the vaccine administration, we're really excited, has started on campus. And so, you know, we really want to acknowledge Dr. Miller's work, that they are doing 200 vaccine administration. All that we get, she is giving on a weekly basis. In fact, she can do this in one day, get all 200 out. And John McMillan's on the line if people have questions about how that's all happening. Okay, and then just finally to reiterate, these are the things that I think we need to be doing to keep us protected. We need to constantly be wearing masks, ideally N95 or double up on your masks. What we're seeing with these new variants is with the increased transmission ability, you need to increase your protection. So definitely wearing, if you can double up on masks, if you don't have access to N95s, I think that's important. We need to continue this mass testing. I would love if everywhere had the testing ability that we have on campus. I think that would be really helpful. Ideally, we would love to see more at-home testing that is cheap and affordable. There has been the first at-home test approved by the FDA, but again, supply is a problem. And surveillance, both the serological, as well as I know there are some labs at UCSC that are doing viral sequencing. We need to continue that effort and to be able to spot these variants as they're coming out. And our UCSC genome browser is doing a great job of collecting all of those sequencing data and making it available so that we can keep track of the variants. Okay, and then very quickly, I want to address some conspiracy theories because our hope is that you will all go out there and help advocate for getting vaccines out there. And so first is that neither COVID-19 nor the vaccine impacts fertility. There have been some conspiracy theories out there that the spike protein looks like another protein that's important in placental formation. But there, we know that there can be short stretches that look similar in any protein you take, right? And so I just want to emphasize the fact that what we're seeing is COVID-19 has been around for over a year. There is no impact on the ability to get pregnant or sustain pregnancies. And so I want to say that there is no concern that the antibodies that are being generated from a COVID-19 infection or from a vaccination is in any way impacting people's ability to conceive. And the mRNA and adenovirus vaccines, these are not gene editing. The RNA and the DNA are transient. They are there just to spark that immune response, get it going, and get your memory. And so these are short-lived and are cleared from your cells pretty rapidly. And then finally, there are no tracking devices planted in vaccines. And when I heard this one, I immediately thought of the fact that we all have cell phones that are easy tracked. We don't need to do anything else. And so there is no tracking devices in vaccines. And let's open the floor up to any questions. Oh, Susan, I see a hand up.
Yeah, that was great. I learned so much, and I wanted to mention, in part in response to Ortiz's question, he said, are there any plans to change the messenger sequence in the vaccine to deal with these variant strains? So Melissa Moore at Moderna is giving a seminar here in RNA Club and OSTEM. She's giving it March 4th at 4 o'clock, so put that on your calendars. I heard her give a talk this past Monday to a different group. It was really great. And she said they may be having to advocate for a third booster to try to deal with the variants. And that another potential prediction is that we will need to be vaccinated against this yearly, like we are against influenza. So those are two of the many, many lessons that I learned from her Monday talk. Thank you for doing this. This was great.
Right, that's correct. I think many of the companies are developing what would be considered a booster shot that are adapted for the new variants to make sure they would stimulate immunity to the different variants.
Yeah, and I think what's kind of cool is the fact that with the mRNA technology, you can do this pretty rapidly, right? So it's kind of amazing that you can get those boosters in place. They're already making them.
There's a question from Barry about, can you say more about the RNA in the vaccines? Is it for the whole coding sequence? Yes, including the transmembrane domain of the spike. And what is the region on either side of the co-signing region? There is an untranslated region and there's a poly A tail. The mRNA is capped. I'm not exactly sure about the specifics of the regions outside of the coding region. Do you have any more to say on that?
That's all I would say.
Okay. Melissa, wouldn't sequence variants require new clinical trials? That's a good question. I mean, we don't need clinical trials every time we update our flu vaccine, but you would want to have some sort of proof that the booster does protect against the new variants. So I imagine there would be some sort of clinical trials done with that.
Okay, so I thought I heard reporting that there is significant antibody response 14 days after the first mRNA dose, but I don't see that in the slide that you presented, Rebecca.
Right. There is, what I chose to present was the neutralizing antibody levels, which is different from the total antibody levels. So you do see significant antibody responses. So I think just a little bit different scales based on neutralizing antibodies versus total antibody response.
Yeah, and Samantha, I'll address your questions. You're asking how do current monoclonal antibody treatments for severe infections affect vaccine effectiveness? And that is a great question. So yeah, you will be asked if you received a monoclonal antibody for severe COVID, you have to wait at least 90 days before getting the vaccine, because there is concern that those antibodies could impact the effectiveness. And so by waiting those 90 days, then an individual is cleared to be vaccinated.
Okay, from Sophie. On TWIV, I heard that if it continues to be an mRNA, multiple mRNAs, sorry, maybe she's just answering from someone else.
Sorry, so I heard that if Moderna, say, just goes with a single mRNA that is, now has the new variants in it, they would not need to do a new clinical trial. But if instead they decide now we're going to have, multiple mRNAs packaged into our particle, they would have to do a second clinical trial.
Great, thank you. And yes, shout out to TWIV. This Week in Virology podcast is really great.
Francisco's asking, how long does immunity last in natural infection? Do we have an idea how long immunity would last with COVID vaccines? Yeah, so recent data from Shane Crotty's group at UCSC is showing that, 8 months out, which is the longest data they have, that there is still strong protection following infection, which is pretty promising. And I guess from what we know about coronaviruses in general, the estimate would be 1 year, 2 years in terms of ability to protect. And we just don't have the data yet on vaccines as to how long the protection will last. So we're hoping the same, at least, if not longer. And Ortiz is asking, does the gold standard qPCR test amplify the spike RNA? Would someone vaccinated with active spike mRNA in their cells test positive with this test in a certain time frame? I would say that you wouldn't test positive in terms of, the vaccine will be peripheral and you're going to take the qPCR from the nasal passages. And yes, the qPCR, as I understand it, is to the spike protein. I believe, unless someone else can clarify that, because I do, yeah.
There are multiple probes. I bet some other people on this call could say exactly where the probes are for the qPCR, but I agree. I wouldn't expect the mRNA produced in your cells in your arm to be able to show up in your sample.
This is Elena. Yeah, so actually for the qPCR, it's against the N. So somebody said in the chat, the nucleocapsid.
Right. Okay.
So yeah, so the spike would not show up. We've had vaccinated people get testing and they don't show up as positive.
Perfect. And that makes sense, because I did wonder if you're having a lot of mutations that your qPCR would no longer be effective if it was to the spike, for example.
Right. Okay, mRNAs use, yes, 1-methyl pseudouridine modification of the RNA, and that was shown to, it increases stability of the mRNA, improves expression of the protein, and I think maybe stimulates less the innate immune receptors.
Exactly.
Okay, is there any knowledge for priority for vaccination administration on campus? Timeline for grad students on campus? And this is a question for John McMillan.
Yeah, so the phases for on campus are laid out in an email from Vice Chancellor Latham a few weeks ago, and it's on the Student Health Center website. We are actually ahead of the curve compared to almost every other campus. We've already worked through group 1. We're mostly through group 2. And group 3 will include graduate students that are working on campus. However, I will very strongly say all of that is subject to change at a moment's notice. I don't know if anybody saw the Sentinel this morning. There's an article about groups that are moving faster than what the county priorities are moving, whether they will put a slowdown on those. So if we're moving too fast, they might actually say we can't move on to the next groups yet. So there's always that possibility. The state changes their guidance on a seemingly daily basis, so we have to respond to all of those things. I can tell you though, I've advocated that all people working in labs on campus be at the top end of the group 3, and there seems to be good support for that if they do it in a tiered fashion. But it's unclear because we have to follow UCOP guidance on that to some degree. But we're definitely pushing and advocating for students and others doing work on campus to get that done as soon as possible.
Now, there was a COVID forum this morning also in which this vaccination issue was discussed. And basically they're saying week to week, we don't know how many we're gonna get. We got 200 last week, we got 200 this week, and some of us over 65 got our first dose. They really don't know. It's a week to week thing, whether they're gonna get another 200. And that's why it's so hard to predict who's gonna get vaccinated when.
That's correct. And the UC, we get all of our allocation from the UC system where we're part of a multi-county entity. And the UC system gets allocated actually a fairly small number of vaccines. And then there's a formula for how each campus gets their allotment.
And Anna asked if someone who has had COVID, would they expect to have a negative response to a first or second dose of the vaccine? And what is being put forward now is that you would think that if you've had COVID, that by you might only need 1 dose of a vaccine because it's going to act as a booster. And so you already have some immunity. And so getting that vaccine is going to help increase that immunity. So I think you won't have a negative response. It will just be a booster response. And maybe before we finish, I think both myself and Rebecca would just like to say that we are happy to be a resource to try and answer questions if anybody has them, or we'll try and find you the answers for things that we don't really know. And so just feel free to reach out to us.
Yes, and I'm happy to stay on and we can keep chatting further. Any questions?
Can I ask one more question? So early on, they were using antibody tests to see how many cases there were above the reported cases. Is there better data now on how many real cases are out there unreported?
I think there's certainly estimations. I don't know if you're referring to our community or Santa Cruz County or just sort of globally.
Well, US.
Right. I'm not exactly sure, and I know Marm had to leave, so he would probably be the best resource for that. I think somewhere around 15 to 20% in the Santa Cruz County area is what he told me recently.
But yeah, I don't think we have active enough surveillance happening throughout the US to really have a very strong answer in terms of, I think in some places it's more than likely been underreported. And Marm is giving a crawl lecture next week. So he is an awesome resource for a lot of the questions.
Right, and you saw that in the chat.
Yes, you
He's
can
really
on the fly, thankfully.
Yeah, yes, really on top of the literature and the especially the-
epidemiological side of things that I'm definitely not as strong. Knowledge base. And yes, we will make the recording available after this.
Okay, Sarah. Anyone else want to chime in with questions or discussion?
I see Maria's question about how do we know how the variants, or do we know how the variants have achieved higher transmissibility?
I think there's, I don't know if there's any
Ongoing.
right.
No worries, thank you. I've just heard like a couple of people not in science asking that question as well, and just being really curious about how this is all, how we're going to handle overcoming that issue.
And I think, you know, from some of the things that I read, it's definitely, you know, you can imagine like the spike protein it's changing in such a way that will make it perhaps easier to be taken up. But exactly the molecular mechanisms, whether it can get into cells that are expressing lower levels of ACE2 rather than having to have an optimal level, I don't think it's fully known, but speculated on that it's everything that's happening is to help it get into cells. And whether there's other receptors out there that we don't even know about besides ACE2 is also possible.
I think it's also has to do with viral loads as well that could be affecting transmissibility. So there is some evidence that individuals infected with these variants have higher viral loads, which may potentially also suggest that they could have more severe disease. But then, so if you're thinking of someone that is infected, breathing, you know, near you or anyone else, if they have a higher viral load, that they are transmitting more virus, right, per unit breath or time, and that it's easier to transmit under those circumstances.
Yeah, and exactly, and we still don't even know that number, like how much do you have to be exposed to, and does that differ from one individual to another?
Yep.
Sophie asked, is there a formal way that we can be advocates for vaccination to the larger Santa Cruz community? And I think just, I know Susan and I have talked about, you know, trying to get out of our comfort zone and being willing to maybe talk with the newspaper, but general public and our friends and family. And I think, you know, explain your enthusiasm for getting the vaccine and being willing to talk about the safety and the thorough, rigorous clinical trials that were done with all of these vaccines, and trying to, you know, just explain in a digestible way the safety and efficacy and how it will protect those people as well as people around them. I did just hear too about the UC Berkeley campus. They're saying people should not go outside even to exercise. And how do we feel about this guidance?
I can chime in because my daughter is currently in the dorms at Berkeley. Apparently they're saying one thing and doing another thing. So they're not enforcing that.
Okay. I mean, first of all, I think it's not surprising that the variant is here. It's, you know, it's just, we haven't had enough surveillance right now to prove that it's here. It's much like the beginning of the pandemic. Of course, we know it's here. And so further surveillance to test for variants, I think is important to guide, you know, public health guidance, but, you know, that is really extreme to tell people not to go outside to exercise.
Yeah. And I think, you know, for anyone who's nervous, that's why I would advise double masking as a way to help protect whether you're outside or you have to go in somewhere. And then Robert asked about vaccine hesitancy is not an issue until there's enough to cover everyone who want it question. I would say, you know, this is why we want to address vaccine hesitancy at all stages, because, you know, there's definitely concern that when you start a vaccine program, say for over 70s, over 60s, you know, they start at that age because they're at most risk. And so anyone that's hesitant in that group, we would like to, you know, alleviate that hesitancy so that they are taking the vaccine as it's becoming available, and not waiting. Is Rebecca's serological test available anywhere?
We are just finishing up the experiments to apply for FDA approval of our test. And hopefully once we receive that, we can start implementing it. But how that is implemented and put to use, we're not sure yet. I do wonder, as Susan mentioned a little bit earlier, if there will be a policy to test for people that had post-COVID infection or are already seropositive, that they would only need one vaccine to be covered. And so serological testing can be useful to implement that, but there's no FDA guidance on that yet. All right, well, maybe we'll just say thank you to those of you who are left, and thanks for all the feedback and comments. And I'll just say again, I think reach out to us if you guys have any questions and we'll find the answer for you.
Definitely. Thanks, everyone.